share hers. So Roxanne is an environmental health scientist working at the intersection of aquatic ecosystems and human health. She has expertise in mercury, other heavy metals, and important nutrients. Roxanne received a bachelor's degree from the University of Pennsylvania and a PhD from Dartmouth, where she worked on the interdisciplinary Superfund research program. She is a faculty member at the School of Marine and Atmospheric Sciences at Stony Brook University. Welcome, Roxanne.
Thank you, Juliana, for that introduction. And thank you, everyone, for being here. I know it's a busy time of year, so I appreciate you taking the time. And I'm going to try to make it worth your while. I'm actually really excited about the results from this project where we set out to try to understand mercury patterns in Lake Champlain in wintertime compared to other seasons, and to see how might that impact our environmental health risks now, but also in the future. Okay, so I'm just going to get right into it. Why do we even care about mercury? It's an important background to keep in mind. It's a global and persistent pollutant, so it's not going anywhere anytime soon. It's a well-known neurotoxin that primarily impacts the central nervous system, but there's also evidence that it can have immunological as well as cardiovascular effects. And in the environment, it can occur in multiple chemical forms, two of which are the organic form of methylmercury, and then there's also inorganic mercury. And the difference here being that this methylmercury has this methyl group attached, and that's all it takes to make a huge difference in terms of its characteristics, including in its bioavailability. It's much higher, meaning that it can be taken up more readily and incorporated into the body. In terms of biological half-life, it's also a lot longer. Oops, I'm sorry. It's also a lot longer for methylmercury. And that means that once it's in the body, it's retained for a longer period of time. In terms of toxicity, it's also much higher for methylmercury than inorganic mercury. And these characteristics have been demonstrated at all levels of the food chain. So overall, methylmercury has this higher tendency to bioaccumulate or increase in the body over time, biomagnify or increase in concentration through the food chain up to predators, and reach toxic effects. And most people are exposed to methylmercury from food, and in particular from fish. And so scientists have studied mercury in fish and also mercury in lakes for decades. So this is a figure from Web of Science showing results of publications if you use the search terms mercury in fish or mercury in lakes. And you can see that since about 1990, there have been over 5,000 publications with these search terms and over tens of thousands of citing articles. And about in the mid-1990s, some seminal papers started coming out that really described mercury bioavailability, biomagnification, and trophic transfer very well. So we've done a lot of work on mercury in fish and in lakes. But there's still a lot that we don't know. And in particular, a lot of field studies that have been done have been conducted in spring, summer, and fall, but not in winter. So these processes of bioavailability and accumulation have really been limited to just those three seasons. And so here's an example of a study done on Lake Champlain by Celia Chen, who's a colleague of mine at Dartmouth College, looking at mercury levels in zooplankton in Mallett's Bay and Missisquoi Bay. And so you can see the methylmercury levels on the y-axis going from about June into October. And one thing you can see is that the mercury level in Mallett's Bay can be higher in zooplankton than in Missisquoi Bay. And you see a seasonal pattern too, where in Mallett's Bay, it peaks somewhere in the summertime and then decreases in the fall. It also decreases in Missisquoi Bay, but the pattern is different between these sites. And so these studies are really valuable because, first of all, the technological challenge of measuring trace amounts of mercury in small organisms at the base of the food chain is very challenging and difficult to achieve. But they also shed a lot of light on the mechanisms of why we see these seasonal differences and site differences. And those can be due to things like differences in phytoplankton or zooplankton biomass, or like turnover or mixing that can be seasonal. and also in methylmercury production and cycling. But again, what's missing is what's going on during wintertime. And so a little bit of background about how methylmercury is produced. It's produced primarily in the sediments in lakes, and bacteria convert mercury to methylmercury under anoxic or oxygen poor conditions. So those conditions are really important for methylmercury to form. And then once it is formed, it becomes available for algae to take up directly into their bodies and then transfer through the food chain, where it can build up in organisms over time. And the conventional wisdom was that production of methylmercury can vary seasonally, and that also the production and the bioaccumulation that results from that is relatively negligible in winter because it's just simply too cold. And there was one study that showed that, but since then, there's been some recent evidence of different types of biological activity in lakes that occur during winter that could support methylmercury production and bioaccumulation. So specifically, some research has shown primary productivity or algal growth that can occur during winter. And if algae is growing, it can take up methylmercury into the cytoplasm. There's also evidence of zooplankton growth during winter. And if zooplankton's growing, it needs to be eating food. And if it's eating food, it's also consuming mercury from that food. And importantly, too, there was evidence of microbial activity and oxygen depletion under ice. So in the benthos or in the sediments where oxygen levels would decrease under ice. And this has been shown by Andrew Schroth, who's also an investigator on this project, using data from actually Lake Champlain. And as I showed in the previous slide, these functions are conducive to methylmercury production. So we expect that maybe based on this, methylmercury is available during wintertime, but it hasn't really been looked at. And this is really important to understand because it would allow us to understand year-round bioaccumulation, trophic transfer, and then risks to wildlife and humans who eat fish. But there's also potential sensitivity to climate change. And there's a lot of climate-driven changes that are expected that could impact mercury patterns in lakes. So for example, increasing lake temperatures and flooding, but then we also could have indirect effects from changing lake biogeochemistry, lake mixing or turnover, and the ecology. So just the species in the system can change due to climate. So the overall effects of climate on mercury in the future are unknown, but likely to be complex. And I think particularly relevant to this project is what are going to be the effects of decreasing ice cover. So here's a figure showing the timeline of ice closure on Lake Champlain going all the way back to 1816. And what you can see is that the frequency of years in which the lake did not freeze over has increased recently. And also since about the 1950s, Lake Champlain has begun freezing later in the year, so the duration of ice cover is lower. So we had two goals for our project. One was to first characterize winter mercury patterns compared to other seasons, and second, to predict risks of exposure based on a changing climate. And so I'm going to only focus on this first goal for today. And there were two questions related to that goal. First, how does mercury supply to the food chain in winter compared to other seasons? And to address this, we conducted a field study in 2 Lake Champlain basins. And second, what's the extent to which anglers rely on eating fish caught from Lake Champlain in different seasons? So to address this, we conducted a survey to assess fish consumption in both recreational and subsistence anglers in the Lake Champlain region. And so I want to tell you a little bit about our field sampling sites. We chose 2 eutrophic bays on the north side of Lake Champlain, Missisquoi Bay and St. Albans Bay. And the reason for choosing these is twofold. One is that these were existing monitoring sites in previous studies where the physical, biogeochemical, and ecological factors were well measured year round, so including winter. Also, they're both eutrophic. So here you can see this red area for both bays shows the excess phosphorus in each bay. So they're both eutrophic, they're both very productive in terms of algae growth. And eutrophic systems like this are becoming increasingly common in the US and worldwide. So it's really important to try to understand those systems and also recognize that there might be some variability just within eutrophic systems. And in fact, these two bays can differ in a number of factors. First, Missisquoi Bay, you can see, has more phosphorus, it's more eutrophic. And based on that, it has a higher potential for lower oxygen conditions. It's also more shallow, and it has more regular cyanobacteria blooms. It tends to also get a lot of water input from the surrounding watershed, whereas St. Albans Bay tends to get water exchange with the inland sea and the northeast arm. Missisquoi Bay also freezes earlier and more consistently than St. Albans Bay. So our hypotheses were pretty simple. We wanted to see, is methylmercury available in winter? Can we measure it to be above detection level? And secondly, we wondered whether methylmercury levels could increase through the winter as anoxia develops under ice cover. And I wanted to put this great picture of Vivian Taylor from Dartmouth and Saul Blocher from UVM conducting some of the sampling during winter, because it's important to point out that this is challenging. You know, there's a reason why we don't have a whole lot of winter data, and that's because it's just challenging to collect. It's cold, it's unpleasant. You have to drill these ice cores and collect samples from there. Your gear can malfunction, and it's less safe. You have to make sure that the ice cover is thick enough to safely go out on it. So to measure methylmercury supply in the water column, we collected water samples at the surface of the lake and near the sediments. We filtered the water in the lab, so we were able to get a dissolved concentration, and that's really measuring unbound mercury in its dissolved phase, but also a particulate concentration, which is measuring mercury bound to suspended particles, whether they be phytoplankton or non-living matter. We also measured mercury in mussels and sediments. I'm not going to get into those data today. There's just simply not enough time, but those will indicate bioaccumulation and storage of mercury in the sediments. And we measured a number of different water quality factors, including nutrients, temperature, oxygen, and ice thickness. And so I'm going to give you kind of an overview of our results. So this is a figure that compares the dissolved concentrations of methylmercury in the water column across different dates, going from September into the winter time, and then in the summer and fall of the next year. And it's methylmercury in the dissolved form for Missisquoi Bay and St. Albans Bay. And the surface samples and bottom samples, the surface samples are in the open circles, the bottom samples are in the closed circles. Those are pretty similar in terms of mercury concentration, so I've combined them for simplicity. And what you can see is that the samples collected during the winter time were certainly detectable and within the range of what we see for the rest of the year. We also get significant differences among seasons. And in Missisquoi Bay, the dissolved methylmercury levels are actually highest in winter compared to other seasons. So that was surprising. But in St. Albans Bay, they're lowest in winter compared to other seasons. So you see these very distinct patterns between these two bays, even though they're both eutrophic, they're both in Lake Champlain. We get higher mercury concentrations in the dissolved phase in Missisquoi Bay overall compared to St. Albans Bay. And this is consistent with lower oxygen levels that we observed in Missisquoi Bay that's conducive to that methylmercury production in the sediments. We also observed thicker ice cover in Missisquoi Bay, and that could probably contribute to the greater differences in the oxygen levels that we observed between bays, especially in wintertime. Now, if you look at the particulate phase methylmercury in Missisquoi and St. Albans Bay, we see similar concentrations overall between the bays. We also got significant differences among seasons. So these letters, if you see a time point that shares a letter with another time point, that means they're similar. If they have different letters, then they're different. So we did see, again, significant seasonal differences, but more overlap, particularly in Missisquoi Bay, than we saw with dissolved mercury. We still found, though, that winter levels were detectable and within range of the rest of the year. And we see similar concentrations in wintertime in Missisquoi Bay compared to other times of year. But in St. Albans Bay, we see something a little bit different. We get the highest mercury concentrations in winter in St. Albans Bay. And so that was very different compared to the dissolved methylmercury phase, which were lowest in St. Albans Bay during winter. So what's going on here? Well, this is strongly negatively related to total suspended solids, especially in St. Albans Bay. And this is consistent with biomass dilution. So what that means is that when you have in the summer months a lot of algal biomass growing, that can dilute the amount of mercury that is bound to that particulate matter compared to when you have less phytoplankton in the system in the winter months. So overall, we found some clear evidence of methylmercury supply in winter. Both the dissolved methylmercury and particulate mercury can be highest in winter compared to other seasons. And this is interesting because it contrasts with conventional wisdom. And so we need to think about what this means in terms of risk. The differences in patterns that we observe between bays, both in the dissolved and particulate phase, really depend on the conditions. So in Missisquoi Bay, we see dissolved methylmercury highest in winter. And in St. Albans Bay, we see dissolved methylmercury that's lowest in winter, but the particulate mercury is highest in winter. And this can be related to things like oxygen level and suspended solids. We did not see any consistent accrual or increase of methylmercury under ice cover. But we're starting to see some potential factors emerge that explain the seasonality and also the differences between bays. So including oxygen, ice cover, and total suspended solids, as I mentioned. But there are also lots of other factors that are likely to have a role: phytoplankton biomass, nutrient concentrations, and temperature, for example. So now I'm going to switch gears a little bit to shift to that second question where we're looking at fish consumption patterns in anglers in the Lake Champlain region. And so just like mercury in fish and mercury in lakes, mercury risks to humans have also been studied for decades. And some of the more recent work has looked at balancing the risks of mercury exposure and also exposure to other contaminants with benefits, because of course we know fish is a really important source of lean protein, omega-3 fatty acids, and even things like vitamin D. And so as a result, fish consumption advice is becoming more complex, and discrepancies among government agencies and nonprofit organizations that dispense this advice can create some confusion. So that's where some of the work there is going. But despite all of that, we still have this knowledge gap where there's very little known about subsistence anglers. So these are individuals who rely on fish that they or someone they know caught for food. Particularly in the Lake Champlain region, we know little about subsistence anglers. So we don't know much about fish collection and consumption habits or their need for informational resources or even access to healthy fish. And so because we know little about subsistence anglers, they are potentially underserved by our efforts to create consumption advice. So to try to address this and try to fill that knowledge gap a little bit, we conducted this survey, and you can see it here. And this was an online survey targeting anglers in the Lake Champlain region. And we took this flyer and advertised through fishing community forums as well as fishing sites, including boat launches and piers. And our goals were to assess local fish consumption patterns and the needs of subsistence anglers, but to try to gain that information that could ultimately inform risk so that researchers and managers and even the communities themselves can provide more resources for subsistence anglers. But this can be challenging. So subsistence anglers have been studied in other regions. Sometimes they're rare among anglers in general. And if they're rare in the Lake Champlain region, the challenge is, can we reach them? And we found in our survey that subsistence anglers were rare among respondents. So we had our survey open for 2.5 months in the summer of 2021. We got 278 responses. And we asked the question, why do you go fishing? Select all that apply. And 42 or 15% of the respondents said that they fish to feed themselves or their family. And so for operational purposes, we're going to call these subsistence anglers. We also had 232 or 83% of respondents who didn't say that they fish to feed themselves or their family. So for operational purposes, we'll call this group recreational anglers. And so this definition, it's important to point out, is not perfect. It doesn't assess the reliance on fish that subsistence anglers catch as a food source compared to other sources of food in their diet. But it's still a necessary part of that definition and could help shed light on some of the patterns too. And so here's just a summary of comparisons between subsistence anglers and recreational anglers based on chi-square tests. We found a lot of similarities between these groups. First, most of both groups commonly catch and eat trout and perch among the fish types. But it's important to note, especially for people who might be listening that are doing their own studies of subsistence anglers, we didn't include a complete list of all the species that could be caught in the Lake Champlain region. So for example, smelt and other things. And you might want to consider doing something like that, particularly if subsistence anglers might eat different types of fish than recreational anglers. For both groups, most or 80% are aware of consumption advisories. So I think that that's good news. Of course, I always think about the 20% that aren't aware. We can't forget about them. They share their preferred source of information for these advisories. So that includes bait and tackle shops, signs at fishing areas, and government agencies. We found no differences in the frequency of the parts of fish consumed, so muscle, skin, and organs, with muscle being the most common, either as a fillet or steak. They also had similar frequencies of fish consumption, with most of both groups eating fish about once a week. And we found similar distribution of ethnicities, race, and the highest education level. So where we did see significant differences is for the age group of 18 to 34-year-olds, we had a lower than expected frequency of respondents of that age among subsistence anglers. And we had a higher than expected frequency of subsistence anglers with an income level of less than $35,000 a year as their household annual income. And this, I think, is important because this lower income group is understudied. So some of my work and that of others has found that higher income is typically associated with higher methylmercury exposure. But now you also have this lower income group, particularly with subsistence anglers that could have higher mercury exposure that is just not as well studied. So we also found clear differences in seasonal fishing habits among these groups, where a greater percentage of subsistence anglers go fishing often in each season compared to recreational anglers. But statistically, we can see that in particular during winter time and during spring, these arrows are showing what's statistically significant, that there was a significantly higher percent of subsistence anglers who fish often in spring and in winter than expected. And when I showed these results to some colleagues, I learned that this is consistent with indigenous fishing practices, where fish caught in winter are actually preferred. They're firmer and taste better. They're easier to catch. In particular, this is true for smelt. And some people will freeze fish in the lake water that are caught during winter and store them so that they can eat during other times of year. So that kind of indicates the level of preference for winter fish. And it may not be true for all subsistence anglers, but it certainly merits further looking into. So subsistence anglers catch and eat fish in the winter more often than recreational anglers. And I want to point out too that these surveys have limitations. It's self-reported information, yet nobody has perfect recall in terms of their diet and their fishing habits, right? We also have low sample sizes. So all of this, I think, needs confirmation with other studies. And I also think that interviews and working with specific communities that could include subsistence anglers within them is likely to yield more insight. And so I'm glad to be working with R.A. Chiapella and Bindu Panikar, who have a Lake Champlain Sea Grant funded research project that's doing just this. Okay, so some conclusions and next steps. So we found that methyl mercury supply in the water column is certainly sustained through winter, and in fact can be higher in winter compared to other seasons. So we have this potential for year-round trophic transfer, but we haven't incorporated this information into our models yet to understand the impacts in trophic transfer and exposure to people and wildlife who eat fish. And also, this mercury supply, the way that we measured it, has been correlated with mercury levels in zooplankton and in fish. However, there are a lot of important factors and lots of steps in the process to get from the mercury supply in the water column into organisms and into humans. So it's likely to be not a simple story. And what we have up next for our project is to also look at mercury in the sediments and in mussels to get a better picture of storage and bioaccumulation respectively, and also look at relationships with nutrients relative to some of the other factors I talked about today. And we'll also look at model predictions of mercury transfer under different climate scenarios, because I think the question is what happens in the future if wintertime looks a little bit more like springtime or a little bit more like fall? What will happen with fish mercury levels at that point? And this, the other big thing that we found was that subsistence anglers consume more winter caught fish, more often at least, than recreational anglers. So the question is, are subsistence anglers at a greater risk of mercury exposure due to that winter fish consumption? And I think beyond our project, there's a need for future work, specifically more extensive study, looking at other types of ecosystems and in context with other factors, focusing on this wintertime process. And there's also a need for more intensive study on winter patterns themselves. So studies that could do more consistent sampling and more intensive sampling, which is challenging to do. Like I mentioned before, it's challenging work, and just to make sure the conditions are just right so that it's safe to go out there. But I think that will be important. And so the most important slide, I want to thank the collaborators on this project, Andrew Schroth, Vivian Taylor, our outreach collaborators, Chris Stepanuk, who helped a lot in developing and distributing the Angler Survey, and Maureen Murphy at Stony Brook, and of course our research support from Saul Blotcher, who helped in the field, and Celia Chen, and our funding from Lake Champlain Sea Grant and Stony Brook University, which of course without, we would not have been able to do any of this work. So we're really grateful for that opportunity. And please, if any of you have any questions or want to talk more about this, please feel free to contact me. And that's it. Thank you very much.